Okay.
Take second.
Yeah, you will have to download.
I'll just download it now.
Yeah, do that. Okay, so
What
I
the?
will use what? You will look for V transfer. Go to your mails and look for V transfer. Like not a V, a V.
Just a second. Let me just download. So basically, while I was doing this, I was also looking at this thing called Cambly, if you've heard of it.
Like what?
There's like this English teaching thingy website.
Okay.
So it's called Cambly. And I applied for some reason. That reason being I want money throughout the summer.
Okay.
So I did that.
Yeah.
So while I was looking at your things, I like the first email, I didn't see yours. Also because I saw that email and then I realized, oh shit, your email is also here.
So what happened to that teaching thing? Is it working? Are you going to do it?
Depends on whether they when they activate my profile.
Oh, okay. If you are looking for some teaching gigs, then there are certain kids who I know who are within the grades of 3 to 5 and are looking for somebody to teach them science, math.
I'll do 7 to 8 month.
History.
Those I can do.
7 to 8 don't want any tuitions. They don't want to study.
Yeah. But 3 to 5.
These are the transition periods for the parents. Most of the parents would transfer their kids from one school to the other when the kids are between 3 to 5. So that's when the tuitions come in.
Okay. I don't know, but 3 to 5 seems a bit young to me.
Yeah, but those are the malleable ages, so you can form some learning habits with them,
Yeah.
So there are several kids that I have who've been inquiring for classes and I've been continuously telling them no, but I've also not been able to refer them to any other person because
I'll now 20th जब मेरे exams 21st को हो रहा and then we're going out till the 30th. So if it is possible, I can take from June 1st, but I'll have to let them know.
They have exams in June. They have exams coming up within a week. They want when the schools are on, not when the schools are done.
But June में school शुरू होता है ना?
अभी तो बच्चों के एग्जाम्स चल रहे हैं ना ऑनलाइन.
Oh, अजीब.
Yeah, so they don't want in the vacations. Everybody wants vacations.
Yes.
Yeah, I know. Okay, so let's go ahead. So what is your cell respiration, or what we usually would refer to as your cellular respiration? Okay, this is going to be something a little more advanced as opposed to what we saw when we speak about your normal respiration that involves your diaphragm getting contracted
anaerobic.
Yeah, all the things that have
simple ass equation.
Yeah, that's not what we are talking about. Okay, when we talk about your cellular respiration, we will take into consideration one singular cell and what exactly is happening within this single cell that is making the production of ATP possible. Okay, now in the last session, what we did is we looked at how your plants are able to carry out this process. In animals, though, this is going to be slightly different. Okay, when you talk about the cell respiration in animals, this is going to be majorly controlling how much amount of energy is going to be produced using your organic substances. So let's say you have a specific amount of organic substance. Using this, what is the usable amount of ATP can you produce is what your cell respiration is mostly concerned with. Now, when you talk about this ATP being formed, With animals, this ATP is going to be the readily available source of energy, or something that is immediately going to be used within the cell. Okay, your body does not want to keep the energy always ready. Instead, what
Okay.
your body does is it keeps a by product of ATP formation, which is ADP and inorganic phosphate, very, very close to each other. And it is only and only through a very small reaction, okay, that your body is able to make ATP from a very portable and very efficient energy source, which is called as ADP. So we are going to look at what are the relationships between formation of ATP and ADP, but you need to
So
understand.
ATP and ADP is basically one phosphate ka difference, na?
Yeah, just one phosphate ka difference. But it
Yeah.
proves to be a very viable method of keeping up with the energy taskings or energy debt, because until and unless you have something ready to go, your body would have to undertake a lot of processes to meet the energy requirements. And in order to
Yeah.
keep this ADP moving into ATP and then using that energy and reverting back to ADP requires organisms to carry out aerobic respiration. Okay, so with aerobic respiration, you are going to be able to yield a certain amount of ATP from glucose. Now,
Okay.
this aerobic respiration, although it is one of the most stronger ways in which body can make ATP, it's not very efficient. Okay, aerobic respiration has certain limitations. You are going to be understanding from this particular process, okay, is that although production of ATP is a very important task, it is not as efficient as photosynthesis. Okay, so we are going to look at aerobic respiration. You need to remember that aerobic respiration merely provides the required amount of oxygen for the process of energy formation. So your aerobic respiration is only and only required to get the oxygen in your cells. So if in the near future you were to find any other method of getting oxygen to your cells, we would not need to breathe. Okay, that's the ultimate goal of science right now, because there's very less amount of usable oxygen in the environment of the earth. Is there any method that we could use to keep ourselves oxygenated without having to breathe the air around us? And if we achieve that feat, well, humanity is saved, but it's a long way to go. Okay, so let's see what.
I mean, why can't we just use the canisters of gas which we have
Your
in the
body needs
hospital?
oxygen, but with pure oxygen, it withers away. It doesn't want
हाँ,
to.
लेकिन वो hospital वाला gas चल जाएगा ना?
But then how? You're still respiring though, right? If you're putting it on your nose, you're still respiring. It's the same thing. We are saying getting oxygenated without having to take in the air through your nose or involving the lungs.
Okay.
That's not possible.
So we want to be cacti, essentially.
Sort of. They also do need oxygen, but they don't have lungs. So yeah, could be. So let's go deeper into what are these respiratory mechanisms and how exactly they work. Okay, if you talk about your aerobic respiration has a very simple equation which has a glucose molecule. You take oxygen. Okay, essentially your photosynthesis reaction reversed is what your cellular respiration is. So if you take a certain amount of oxygen and glucose and you carry out a process called as combustion, what you end up getting are 6 molecules of carbon dioxide, 6 molecules of water, and a very small quantity of energy. Okay, now depending on the resource book you are using, there are going to be variations in the number of ATP molecules that you are going to get. Some resources may say you get 26 ATP molecules. Some resources may say you get 8 ATP molecules. Some resources might tell you that the total net gain is only 2 ATP molecules. All of this is dependent on what kind of processes they are including in the end
Mm.
product of your formation of ATP. We are going to go all the way to the end of this respiration topic, and then you will realize that all of these are actually correct answers. Okay, so how that is
Yeah,
something.
so 2 ATP is in the first stage, 8 ATP is in citrus cycle, 26 is in the electrotransfer chain.
But they say all of this as net gain. That means the total gain is 26, 8, or 2. And all of those options are correct. So let's go.
I
And
don't know, isn't it a cycle anyway?
it is not a cycle. You need every time you need a
Okay.
different glucose molecule to make energy. So it's not the same glucose molecule that gets like broken down. So it's different, but it does make sense. So
But
let's
that
see.
would,
Mhm.
honestly, that would be so cool if we didn't have to have glucose at all.
I know,
Like
but
just one glucose.
yeah, I mean, that would make sense. There are certain animals who eat only once and then stop it. Like your cells do it for a certain period of time. Okay, like if you think of your mitosis, your cells do go through a phase where after a while they stop any metabolic activities. Like if you look at this, and this is your S phase, this is your G phase. During the G and the G2 phase, okay, this is where the growth and metabolism is taking place. But through the entire process of mitosis, your cell actually carries out no metabolic activities, which means during this time, your cell has zero need of oxygen. So, for a period, we do go through this phase. It's just that mitosis is a very short-lived phase of your cellular cycle. So, we can achieve it, but only during cell division. Okay, so like I told you over here, ATP and ADP are two very different molecules, but using them together is going to give you certain amount of energy. So let's understand what and how this is possible. In order to understand more deeply about your ATP, you need to know that ATP is adenosine triphosphate. Adenosine or adenine, right, is one of your nitrogen bases that make up your DNA base pairs, which means that it is going to be a structure that is widely available in your body all the time. So this is what an adenine base looks like. To the adenine base, you have the ribose or the pentose sugar that is going to be attached. And it is to this ribose sugar through a squiggle bond. Okay, I'm going to write this over here. This
What's
is
a
called
squiggle bond?
squiggle. So a squiggle bond is a bond where you have excess amount of energy going into making this particular bond. Okay, the squiggle bond is something that stores excess amount of energy. Okay, and this excess amount of energy is only and only going to be available when the phosphate molecule is going to end from this particular position. Okay, and you are going to use this energy from your squiggle bond to just make ATP molecule. That's what a squiggle bond means. Okay, and this is it's shown by squiggly lines. That's what is important.
Okay.
Okay, so you have 3 squiggle bonds that go into making adenosine triphosphate. Okay, so you see this is adenosine triphosphate. Uno, dos, tres. Okay, so triphosphate is what you have. Now, every single time that you go on to break one squiggle bond, okay, you can call the squiggle bond also as a very powerful bond, a bond that has
Okay.
an amount of energy in it. So when I go ahead and I break this bond, okay, between two phosphates, as a result, what I do get is a certain amount of energy. So the energy is not because of the ATP molecule, but because of the breakdown of ATP into Now this structure till here would be my ADP. This is adenosine 1, 2. So
Okay.
diphosphate, right? And adenosine
Okay.
diphosphate has lower amount of energy within it now because this amount of energy just got released,
Okay.
right? So that's something which
Yeah.
you need to remember, and you need to understand that every single time that you are going to break these bonds, okay, every single time that a squiggle bond is going to be broken, it releases the energy, and the energy released by the first bond is higher than the second bond and lowest in your third bond. Okay, that is another very important part to remember. So every single time that you are going to break these bonds, certain amount of energy does get released, and it goes on decreasing as you go on making more and more smaller phosphate molecules over here. That is really important to notice. Also, you can break another bond from here and go on to convert your molecules into AMP. These AMP molecules also have a significant role to play in your what we would call as your metabolic cycles. Okay, but these AMP molecules are very rarely going to be made from the breakdown of ATP. Your body is capable of synthesizing its own AMP molecules without ever having to reach out to ADP or ATP for its formation. Okay, so AMP molecules do exist in your body, but they are not very specifically required to be made from ATP. But all the ADP molecules, yes, they are going to be a byproduct of ATP breakdown. Okay, so this is something that you want to remember. You want to understand that this is the relationship between your ADP and ATP molecules. Now let's go
Okay.
over and understand what does this information help us with. Now you know how ADP and ATP are going to be made. If you take a small quantity of glucose, at this point I'm going to start off with just one 6-carbon compound, one glucose. I know that as a reaction to break down this particular molecule, I will have to take up several step reaction. Okay, it's not a single step
Okay.
or two-step reaction. Glycolysis is a several step reaction, at the end of which I end up having two, three
10
carbon
ATP.
compounds, right? I end up having
Okay.
something called as pyruvate. So this is the guy,
Okay.
pyruvate. Okay, so I take 6-carbon compound, and then I end up making this molecule called as pyruvate. Now, it is very important that you remember the structure of pyruvate. It isn't very difficult, but it is essential that you remember it. Okay, so your pyruvate looks like this. Okay, this is the base structure. It does have a several number of molecules attached with it. So you are going to have a double bonded O here and a double bonded O here, O and CH3. This is what your pyruvate molecule is going to look like. Okay, this negative charge represents that another molecule can come and attach over here. So this is not a stable molecule. Pyruvate remains unstable and has to be put into a new cycle or into a new reaction so that it can be used up. You cannot at any point of time leave pyruvate on its own because it is unstable molecule. So it enters into a new cycle right after glycolysis, which is called as Krebs cycle. But before we go there, we need to understand things about your glycolysis. So this is the process. Okay, glycolysis is where you are going to break down glucose, which is a 6-carbon compound, into two 3-carbon compounds. Along with these two 3-carbon compounds, you make two ATP molecules. The net gain through your glycolysis is two ATP molecules, and you make NADH and a small quantity of NAD plus. Okay, so
Okay.
NAD is nicotine diamide, and you are going to have unreduced or unoxidized precursor. Okay,
Okay.
and you are going to have a reduced form of the same molecule. If I was to draw my glucose, okay, in let's say I have 1, 2, 3, 4, 5, 6. I just said that you are going to end up 1, 2, 3, 1, 2, 3, right? And in between, you give in 2 molecules of ATP. So these are 2 molecules of ATP. You end up getting, so I'll just draw my ATP in a different manner. So 1, 2, 3. This is my ATP. 1, 2, 3. Okay. As a result of these 2 molecules going in, I end up having 2 molecules of ADP. This is the
Okay.
first investment step that I make. Okay, 2 ATPs go inside and I get 2 ADPs out. It is only at the end of this reaction, only when I reach this part, that I will get 2 ATPs back. So this
Okay.
place where I put in the first 2, you would call the first 2 of my ATP molecules, we always refer to this step as the investment step. Okay, here you invest your ATPs. Now, once you have invested these ATPs, you want some returns on them. So what you do is, in order to get some returns, you give a high energy molecule such as NAD plus. And when you give 2 NAD plus, you end up having 2 NADH, which have been now reduced. And then in the payoff of investment of these 2 molecules that have sort of attracted 2 NAD molecules, you end up having 4 ADPs, which will then turn into 4 ATP. But you already invested 2 ATP. So your net gain after reducing all of the investment that you made is only 2 ATP molecules and 2 molecules of pyruvate, which are not so stable, but you got to work with it. You got to run with what you have right now. So let's say now you take these 2 molecules. Okay, so I'm going to just copy this same reaction and we are going to go ahead from here. So I take this, let's select it, copy and paste. Okay, so now this is what you have already carried out. You have 6 carbon compound glucose that has turned into 3 carbon compound, which is known as pyruvate. This pyruvate can have multiple things happen to it. Okay, if this is a animal cell and if this pyruvate is not used up for a very long time, it will turn into lactic acid. Okay, and then lactic acid or your lactate or whatever molecules you have will then have to be entered into different reactions, and then they would be used either for making energy or they'll just probably be lost. But if this was a plant cell, okay, this pyruvate gets converted. So I'm gonna do a green color so that you know this is in plant cells. In your plant cells, this particular kind of pyruvate molecule is going to go on to turn into ethanol or carbon dioxide. Ethanol if this was in yeast, and carbon dioxide if this was in plants. Okay, so this is the fate of your pyruvic acid. This is a question that could be asked to you. What is the fate of pyruvate molecule after glycolysis? So you would have to say these are the 3 byproducts that could be formed because of your pyruvate breakdown. Just give me a second, okay? Okay, so going further, now once we understand, okay, that this is going to happen with your pyruvate, we want to see what exactly happens if you do have the process of glycolysis go ahead. So we need to just simply understand the steps of glycolysis, and that is all that you really need to know. Okay, nothing more in detail, nothing to be understood in like depth over here. You simply need to know what are the different steps that are going to make this pyruvate molecule. So I'm just going to take the same particular reaction that I have. I'm just going to add a few more details, and this detail is the only amount of information that you will ever consider in your answer. So I'm going to use the same annotations as well. So I do have 1, 2, 3, 4, 5, 6 carbon compound as glucose. In order to convert this molecule, okay, of 6 carbon glucose into something called as fructose 1,6 biphosphate, okay, biphosphate. Now see how I have biphosphate written over here. This should give you the idea that I would need to take ATP and convert it to ADP so that I could make this into fructose 1,6 phosphate. Later, I would add one more ATP molecule, and therefore this particular glucose will be turned into a fructose 1,6 biphosphate. Fructose 1,6 biphosphate is a very, very unstable product. Okay, so I'm going to write over here unstable. This is going to be unstable. It is going to form two molecules, each which contain one phosphate with them. So I'm going to label this phosphate with a color. Here and here, okay, and there are two phosphates. So I will have one phosphate here, and I will have one phosphate here, and therefore what I will end up having would be 1, 2, 3, and 1, 2, and 3. Okay, this is called as DHAP. You really, again, do not need to know what DHAP molecule is all about, but if you must, then DHAP stands for dihydroxyacetone phosphate. You are not expected to know this long form. You are not even expected to know what is the process in conversion of these molecules. It just helps if you had a bit of idea about these, so that if they are referenced in your question paper, you would know something about them. DHAP, and you have another molecule which is called as glyceraldehyde 3-phosphate. We have spoken about glyceraldehyde 3-phosphate in our previous reactions as well, right? So here, glyceraldehyde 3-phosphate is the similar molecule that we spoke about in your Calvin cycle, which is a key reaction in your plants. Okay, so you go on to form glyceraldehyde 3-phosphate. Because these two are the molecules formed by breakdown of 1,6 carbon compound, these are reversible molecules. These are enantiomers kind of a thing, and they are going to be similar to each other, and they can be converted from one to another very, very easily. All of this DHAP mostly gets converted to glyceraldehyde 3-phosphate, and a cycle begins which has to be repeated twice, because now you have one carbon compound that has been broken down into two smaller molecules. Okay, so what happens to this 3GAP or GA3P is you are going to bring in NAD molecule, NAD plus, okay, and NAD plus is going to get converted into NADH. You will put in one ADP, and this ADP now gets converted into ATP. And in the last step again, one more ADP molecule gets converted to ATP. And as a result, one phosphate from here is added here. One phosphate from here is again added here. Okay. And now, so I'm just going to make an arrow to show where each one of these is going. So this ended up here, and this ended up here. And so now you have this pyruvate molecule that will enter into another reaction, and you have the first step of cellular respiration being completed. Is there anything that you want me to go over before we move on? Is there anything that you have not understood? Anything that is not making sense? Anything that you feel like, okay, isko ek baar aur discuss karna padega? You're on mute, so if you're saying something, I won't be able to hear it.
I think I get most of it.
Okay. So I'll go on to the next
I
part.
just don't understand how citric acid is involved.
Yeah, so you take your pyruvate here, okay, and if pyruvate is not used up, if this pyruvate molecule does not go anywhere, then what happens is this pyruvate will take in some amount of carbon dioxide, and getting added with carbon dioxide changes your pyruvate into lactate. That's literally all that happens. Okay, so if you just look at the reaction of pyruvate and carbon dioxide, it is very evident that how exactly these two molecules are going to form your lactic acid. Do you want me to write the reaction here so that you could see it? Maybe. Okay, so I'll just tell you what happens. Okay, so let's assume that this is my pyruvate molecule here. Okay, the 3-carbon compound. It has to undergo certain processing. When we say that it has to undergo certain kind of processing, okay, the first important thing that happens is you have this enzyme in your body, which is called as acetyl coenzyme A. Okay, this coenzyme A being added to your pyruvate, okay, kind of takes your pyruvate through a what we call as dehydrogenase formation reaction. Okay, now this is something that you don't really have in your syllabus at all, so it's okay if it all just goes above your head. Just try to understand that when I have the structure of my pyruvate, so I'm just going to draw a very rough structure of my pyruvate here. So I have carbon double bonded to an oxygen and a CH3. Okay, when I take this molecule ahead, okay, so that it could be used up for any other reaction, I would want to remove this carbon dioxide.
Okay.
So I end up having O double bonded to a C. Then this particular coenzyme comes into picture over here. Coenzyme gets added and I have CH3. Okay, this is like a mediatory step between. I have like the formation of lactose take place, because if I just wanted to make a lactose, then the structure would change to be something like this. So just take a look at this structure here. Okay, this is what my pyruvate is going to look like. When I make lactate, my lactate looks like this. There's a double bond O that has gone. I have a C, I have a H, I have a HO, and there is a CH3. Okay, the double bonds are now being broken, and I have added certain amount of hydrogen molecules here. This takes place in your cells when the amount of oxygen is limiting. Only and only when the quantity of oxygen is a limiting factor, your muscles will go on to break the pyruvate into your lactate. This is a very strict stipulated condition. Your body does not like lactate. So making lactate is something that your body has to do in order to tide over the conditions and to make sure that your muscles do not die. So I'm going to write this very detailed, okay, so that you remember this part. This reaction takes place in the cells only when the amount of oxygen is limiting. So only and limiting are the key terms over here. Okay, now once you understand that, okay, so this is basically what is happening in my aerobic respiration. You also would want to know that if you have anaerobic respiration take place, okay, which means then again the oxygen is in limiting quantities, and if this reaction is taking place in your plants, then instead of forming lactate, your body forms ethanol. So again, I have this pyruvate. If I take this pyruvate and I say absence of oxygen, I was able to form again a 3-carbon compound, lactate. But if this was happening in my yeast cells or in my plant cells, then I do have a small gap that occurs over here. And here, what I do have is a release of certain amount of carbon dioxide. And because one carbon is lost here, I form ethanol, which has only 2 carbons. So this is a 2-carbon compound. This is a 3-carbon compound because one carbon is lost here. Okay, this is something that you need to take into consideration. Also, when you are carrying out the reaction or the anaerobic reaction, okay, for breaking the lactose, breaking the pyruvate into lactose, you do take NADH and convert it into NAD. So I'm going to do this. Yeah. NAD plus gets converted to NADH. This is a reaction that occurs in both of these steps. So aerobic respiration requires energy to neutralize itself, whereas aerobic respiration is going to give you the energy. Okay, this is something which is really important. And lactate formation in your muscles is one of the most important anaerobic respiration that takes place in your muscles, and it only helps in maximizing the power of muscle contractions. So your lactate makes sure that your muscles do not go weak or do not go limp because they don't have energy. So lactate maximizes the power of your muscles. This is a very important statement to remember about lactase. So I'm going to put that in a box so that we do not forget about this. Okay, so lactate maximizes the power of muscle contraction. Very important that it helps in contraction of your muscles. Okay, now if we are to understand this, if we say that taking into foods that are going to give you more amount of pyruvate, and then undertaking aerobic exercises is going to make sure that you have ample amount of ATP molecules present in your body, it does imply that you having proper diet and good supply of oxygen is a key to being more energetic. Okay, this is really important. And if you do high intensity workout, the aerobic exercises are going to help you break down all of that energy containing food products such as fats and your proteins into your energy. And this means that if you go on carrying out more amount of aerobic exercises, and if you do certain amount of non-working after a while, then what happens is the lactate levels in your body will increase because now you are not burning those glucose molecules completely. And that means if you do not utilize all of the energy that is to be formed, your glycolysis will stop and it will only give you pyruvate, and pyruvate will give you cramping of the muscles. Okay, so if you do not take your exercises to completion, then they are going to weaken your muscles. It is very, very important that if you start exercising, you do it, you go all the way, or don't do it at all. Don't leave it in the middle. Warm up, exercise, warm up. That has to be the way you go. So now this is what is the background for your aerobic respiration. Another important thing that we have gone over in the previous sessions also is that all of this is taking place inside your mitochondria, right? So all of this is taking place where you do have your mitochondria present. So you have to understand aerobic exercises where you are going to be forming anywhere about 28 to 36 ATP molecules. All of this has to happen within your mitochondrial structures, which means that it is going to be your inner mitochondria, okay, or the inner mitochondrial layer that has to be extra working, or it has to take up extra work in order to make sure that you have good amount of energy being produced through the day. Okay, so if we assume that the glycolysis is something that has to occur, the glycolysis process will take place in your cytoplasm. Okay, because the glucose is available in your cytoplasm. So I'm just going to take this portion and I'm going to color my cytoplasm in a different color so that it is marked differently. Okay, so here you go. This is your cytoplasm. I'm going to write this over here. Okay, and what has happened in your cytoplasm? You took glucose and you broke down the glucose to pyruvate, and you did make ATP molecules. So I'm going to write ATP into 2. Now, in order to get your next reaction started, okay, you have to take all of this reaction material and you have to move it to your mitochondria. So this is going to be your mitochondria. I'm just going to color the mitochondria as well. Okay, so this is what your mitochondria is going to look like. Okay, and it is in your mitochondria that all of these reactions are going to occur. So let's go over what reactions are taking place in your mitochondria. The ATP molecules that you just created, okay, all of these ATP molecules are going to move in, and these ATP molecules are going to be utilized for formation of something. Taking the pyruvate and the ATP molecule, you are going to form something which is called as acetyl coenzyme A. Again, how this is done is completely irrelevant to our understanding. Right now, all that you are expected to know is that in your mitochondria, a reaction which is called as linking reaction takes place. Okay, so this is your linking reaction, or you can also call this as link reaction. It basically means you take the pyruvate, okay, and you convert your pyruvate into your acetyl coenzyme A. If you must know, then this is your pyruvate molecule. Okay, you take the pyruvate molecule and you add a coenzyme A. This coenzyme A goes in. Okay, you have your NAD that turns into NADH. Okay, carbon dioxide is released out, and you have coenzyme A attached to a 2-carbon compound, which is called as acetyl. So acetyl coenzyme A gets formed. This is the small representation of how this reaction is going to take place. Then you have your Krebs cycle that comes into picture. Now, you have been talking about the Krebs cycle since your 10th standard. Okay, so Krebs cycle is a very, very simple cyclic reaction which has to run twice. And after this reaction has occurred, you have formation of two more molecules of ATP that take place. Okay, so this is again a very important process to remember. And if I was to abbreviate this reaction and write it in like very simple terms, then you would have this cycle beginning, okay, with acetyl coenzyme A. So this is going to be your first molecule that comes in whenever you have your Krebs cycle going on. So you have acetyl coenzyme A. This acetyl coenzyme A, so I'm going to just call this as 1. Okay, you are going to form a molecule which is called as citrate. Okay, citrate, give me a second. Okay, so citrate goes over and it gives you one NADH here. You have one more NADH that is formed here. Okay, along with the NADH, you make one molecule of GTP instead of making ATP. You also make FADH. FADH is also similar to your NADH. Okay, it is another energy giving molecule. Instead of having nicotine diamide, it has flavin adenine dinucleotide. So this is your nicotinamide adenine diphosphate. This is flavin adenine nicotinamide, like that molecule. So this F stands for flavin. Okay, there is no major difference between these molecules. It's just that formation of FAD is more easier than formation of NAD here. You get one more molecule of NADH, and this is your one cycle of Krebs. Okay, as a result of this Krebs cycle, you get endpoint. Okay, over here you get one ATP molecule. So this is my one ATP molecule. This cycle runs twice to give me two ATP. So this is one, and when this runs twice, I get two ATP molecules. Okay, as a result of formation of these two ATP molecules, my process of cellular respiration goes ahead and I enter into something which is called as electron transport chain. What happened with your PS2 and PS1, that is photosystem 2 and photosystem 1, is the same thing that happens in your electron transport chain as well. But now what is important to note over here is that in your electron transport chain, there is going to be formation of excess amount of ATP molecules as opposed to what you had in your glycolysis, then your link reaction, and in your Krebs cycle. Okay, so the amount of molecules of ATP that are produced in an electron transport chain are much higher than that of your any of these three reactions. So electron transport chain is a very, very crucial step, and we need to understand this step in detail. So I'm going to go over this very, very quickly. It's very easy to understand. You simply need to have these molecules drawn properly so that you would remember them. So I am going to have my first molecule over here. Okay, I'm going to draw these molecules first, and then you would be able to understand what they are going to do. So I'm just going to make them so quickly. Getting the structures correct is important so that you can put labels to them and you can remember what each one of them is going to do. Structure and location, very, very important. So I have one guy here. I will have one guy here. Okay, and the last one, which is your ATP synthase. Right. Two. Okay, so these are the different molecules that you are going to have in your electron transport chain. These are very, very important molecules. These are all going to be present in your membrane. So you need to remember that all of these are going to play a very key role in making sure that you have the formation of your ATP molecules taking place efficiently. Okay, so you want to remember that this is inside the mitochondria. So I'm going to write mitochondrial matrix in here. Okay, and this is between your two membranes. So this is the outer membrane of your outer membrane of mitochondria. Nothing from your electron transport chain is taking outside the mitochondrial
So,
region. And this is your intermitochondrial space, or you can also call this as intermembrane space. Intermembrane space. Okay. Now comes a very, very important part. Okay. This is something that you have to be able to relate with. Okay. Although these molecules are not drawn to scale and they are not like exactly the way you would want them to look, it still helps if you remember what each one of them is going to do and what exactly are going to be their names. This first guy is called as NADH reductase. So I will just write it here. NADH reductase. And it does exactly what it says. You take NADH and it is going to convert your NADH to NAD plus. Okay. When this reaction is taking place, the hydrogen that is released goes up into your intermembrane space. Okay, this is the first part that you want to remember. Then you have another molecule over here, which is called as cytochrome reductase. So I'm going to write cytochrome reductase. Cytochrome reductase. You have another molecule which is called as cytochrome oxidase. Cytochrome oxidase, and then this is your ATP synthase.
mother,
ATP synthase literally does what it says, takes ADP plus one inorganic phosphate and it is going to convert that ADP and inorganic phosphate into ATP. All this thing is going to be possible only and only when the hydrogen is going to be pumped back in. Where is this hydrogen coming from? So you have one hydrogen molecule that is being released here. You have one molecule of hydrogen that gets released here, and you have another molecule of hydrogen that gets released here. Okay, and all of these hydrogens later get pumped by your ATP synthase, and they carry out the process of your formation of ATP. When you have your NADH getting converted to NAD plus over here, okay, in this particular reaction, another step that takes place is that you are going to have a particular molecule of water that is going to come out here, okay, and it is using this molecule of water that you are able to maintain the concentration of the inner mitochondrial matrix. Without this, your inner membrane will become very, very oxidative, very, very acidic, and you would not be able to, keep the metabolic processes up. This is like what is going to happen. You do have another two set of molecules that are present over here. This is called as a quinoline, okay, and you have another one over here which I kind of forgot to draw, but now that I remember, I will make it here. Okay, this molecule is also very important. It is called as cytochrome C. Okay, so this is a series of molecules that you are going to have in your cellular respiration, and all of these molecules make it possible for the formation of several different ATP molecules that are included in the final, you would say, in the final calculation of the ATP. Okay, now if you have to understand what exactly is happening in your electron transport chain, you would say that as a result of formation of all the energy rich molecules, your body takes in all the ADP, NADH, FADH, and simply converts these molecules into energy rich substances. That is literally all that is happening over here. Okay, if you look at this reaction in detail, although you don't have to remember the entire amount of electron transport chain, because it's not something that your syllabus expects you to know. If you want to go into some amount of detail and understand this, there are several good resource videos that are available, okay, and they will give you a very good understanding of exactly what is happening in order to carry out this reaction. What you are expected to know is that all of these molecules are going to have a very specific kind of function to do. Okay, and all of these molecules are present in the inner membrane of your mitochondria. So if you understand what is happening inside over here, you will be able to explain exactly the process of formation of ATP. I will share one very good resource video with you for this, okay? But when you are looking at that resource video, I would expect you to keep this image in front of you, so that when you learn about the ATP synthase, you understand exactly why this molecule is put at the end. So just give me 1 second and I'll see if I have it already saved.
Uranium.
Guys. You do not need to know the structure as such, but it helps. if you could, relate with this part. Okay.
I have gamified my body. I genuinely believe you cannot improve what you cannot measure.
For the past 3 years, I've been tracking every food that I eat.
Of what's happening?
Yeah, I can hear.
Is it twice, or is it simply once that you are able to hear it?
I can hear properly.
Ever since I've got the CGM, I've stopped that.
Now the aim is I just need to maintain my metabolic score. It helps in work, it helps in personal life as
ATP synthase, a molecular mill. Step 1. Hydrogen ions flowing down their gradient enter a channel in a stator, which is anchored in the membrane. Step 2. Hydrogen ions enter binding sites within a rotor, changing the shape of each subunit so that the rotor spins within the membrane. Step 3. Each hydrogen ion makes one complete turn before leaving the rotor and passing through a second channel in the stator into the mitochondrial matrix. Step 4. Spinning of the rotor causes an internal rod to spin as well. This rod extends like a stalk into the knob below it, which is held stationary by part of the stator. Step 5. Turning off the rod activates catalytic sites in the knob that produce ATP from ADP and phosphate.
So that's the reason why we draw this particular molecule the way we did. Okay, and that's literally all that you actually want to remember. So this is your ATP synthase. It takes in certain amount of hydrogen, which is going to be released by your reductase and cytochrome reductase and cytochrome oxidase, and these molecules are going to release the hydrogen, which is then going to be utilized by your ATP synthase for formation of ATP. That's it. That is what is happening in your cellular respiration. Okay, so I hope you have access to these websites and like note resources. Okay, but I'm still going to share with you one PPT that I have created when I was doing the this particular chapter with my kids in school. Obviously, we took about 11 classes to understand this. We do not have the luxury of using 11 classes to do this. So I have tried to summarize everything in as brief manner, but also so that you understand every specific thing properly. So I have gone over all of these processes, but in order to help you make your answers and in order to write the notes, I am sharing with you the presentation that I had created some time ago. Okay, so I'm sharing with you the presentation. I'm sharing with you these slides. Okay, so starting from cell respiration here till this last one. And I'm also dropping in the links to the videos that I would want you to reference in order to understand this in just slightly more detail. Okay, do not spend too much time looking at the YouTube videos. It's not going to be productive because it is not specific for IBDP biology. These are generalized videos that are going to give you information that you don't even need to use right now. Okay, so just go over just one video at a time and remember what is possible in what I would call as visual manner. Okay, whatever you can remember in visual manner, go ahead, remember it. Rest, I want you to look at the PPT and understand what the PPT is writing, how the answers are framed, and use those words for writing your answers. Okay?
What are the squiggly lines called?
That's just the squiggle bond. It's called as a squiggle bond.
Okay.
Okay, that it does not have any different name. We did not come up with a different name because we already had ionic and covalent and God knows what not different bonds. So we did not want to include another name in syllabus. So we just call it squiggle bond. That's it. Simple, easy, and efficient. Okay, so I have already composed the mail with all of the material. I will just make this into a resource and I will share this as well. And then if you do have any doubts while writing these answers, do tell me and we will
Okay.
get them. So I'm just
By
gonna,
tomorrow.
yeah, I have already removed all the work that you do not need again. So this is a new resource. It is 1 frame per second. Export. Give me 1 second. I will just mail it to you right away so that I do not forget it. I usually do. Okay, so this is the untitled artwork for cell respiration. And followed by that, I'm sending another mail which has the notes and the YouTube video. Okay, so just go over the entire part and you're sorted for cell respiration. Now,
Okay.
the next part that you have mentioned in the message, okay, let me just see, is DNA structure, the structure of DNA and RNA and the nucleic acid. Very, very
Sorry?
DNA, the structure of DNA and RNA.
Uska kya?
You have written that this is the next part that you would want to do. Like your mom sent me the message saying that DNA structure and RNA structure, nucleic acid. So tomorrow we can cover that part.
Okay.
Yeah, and then I have animal physiology. Anything specific in animal physiology, because it's like an entire portion
Thing.
in that. Just
We did neurons a little, but like more on neurons.
okay. Could you write this, because I speak to so
Yeah,
many
okay.
kids every day and then I'm definitely going to forget if you did tell me something. Yeah, so just send me like writing में क्या चाहिए उसमें से and I will just take that off, because it's okay. Ultimately, we want to try practicing more of writing the answers. So, as quickly as we can finish the recap, we can go to solving the papers. Okay. So, ye hai, I'm sending all of this and the video on WeTransfer. Look at the WeTransfer video, download it. अगर तुमने उसको download नहीं किया, तो वो delete हो जाएगा.
I downloaded now.
Yeah, तो अभी जो दूसरा वाला I'm going to send, download that as well. नहीं तो वो delete हो जाएगा. Okay, so this is it. I shall see you tomorrow. Okay.